CT1812 is a small molecule, orally available, and it's highly brain penetrant sigma-2 modulator. And the way CT1812 works is it can bind to the sigma-2 receptor, which then can lead to a displacement of the amyloid-beta oligomers from neuronal synapses. And so this trial, the SPARC trial, read out earlier this year. And what we did there was we collected CSF, cerebrospinal fluid, from patients in this trial were mild to moderate Alzheimer's patients. It was a 6-month trial. And CSF was taken and we did proteomics analysis. So what that entails is that you look at every single protein present in the cerebrospinal fluid of these patients, some of which were given CT1812, others were given placebo. And we did an analysis to determine which biomarkers were pharmacodynamically regulated by CT1812 or altered compared to placebo. And the results were quite exciting. For one, this trial set out to examine a synaptic protective mechanism of action through a PET tracer. And although we didn't present that data at AAIC, that will be subject of a manuscript later this year. What we focused on at AAIC was our CSF biomarker analysis. And it was indeed, the findings suggest that we are impacting key proteins and pathways related to synaptic function, supportive of our synaptic protective mechanism of action. In addition, through unbiased or unsupervised pathway analyses, we identified other pathways that are affected by CT1812 in this trial, and those include the amyloid biology, inflammation, as well as genetic risk factors. We also identified pharmacodynamic biomarkers that we hope to track in our future clinical trials, one of which reads out end of next year, our SHINE trial in a mild to moderate patient population. It's also a 6-month trial. So we would look forward to that reading out and seeing if these findings replicate in that trial. Essentially, as you are likely aware, there are several companies working on amyloid-beta lowering therapies and also alpha-synuclein lowering therapies. So we believe we have a unique approach in targeting the sigma-2 receptor. And really, there isn't going to be a single medicine that cures all patients, right, with this disease. It's a complex disorder. There are multiple cellular damage responses or cellular processes that are disrupted. And so we believe that targeting sigma-2 will be, we're testing it, right? We hope that will be a beneficial therapeutic approach to help patients.